Today we're going to discuss about the biocatalysis involve the structures of enzyme. Enzyme, or another name of enzyme, is called organic catalase or biological catalase. Okay, the function of enzyme is to speed up the rate of metabolic reaction, and in order to do so is to reduce the activation site of the reaction. So why enzyme is an organic catalase? Because they are built up by protein. Okay, they are formed basically by the polypeptide change. Okay, so all the enzyme is globular protein because they are folded in specific spherical shapes. They have the three-dimensional conformations which enables the protein to carry out their specific function, especially the active site of the enzyme, which have the shape or conformation which is fit with the substrate. Enzyme normally is present in tertiary structures or quaternary structures based on how many polypeptide change is used to fold it into a complete functional enzyme. There is two types of binding site found on the enzyme. One is called active site and another one is called allosteric site. We're going to look at how the labeling of the enzyme is done in the next slide. So basically in this slide, we just discuss about what is active site and what is allosteric site. Active site is the binding site specific to bind with substrate. It is a part where the enzyme is bind with the substrate and enable the catalysis process by reducing the activation energy of the reactions and resulted the speed up of the rate for the metabolic reaction catalyzed by the enzyme itself. Allosteric site, which is this part of the enzyme, eventually is the binding site for other substance, which not include the substrate. The binding of the allosteric site, the substance to the allosteric site, normally lead to the change of the conformation for enzyme or the active site. So, depend on what kind of substrate is bind with the allosteric site. If the activator is bind to the allosteric site, it leads to the conformation change to activate the enzyme to carry out the catalytic process. Whereas if today is the inhibitor that bind to the allosteric site, then it will lead to the inhibition of the enzyme whereby the changing of the conformation happen to the active site, which cause the active site to lose the functions to bind with the substrate. So this is basically two different binding sites that found on the enzyme. Now let us look at the second slide, the labeling of the enzyme. So enzyme normally bind with cofactor. Some enzyme normally bind with the cofactor before they're able to carry out function. So before the enzyme bind with the cofactor, we call the enzyme as apoenzyme. So the whole thing here, blue in color, is apoenzyme. They are not yet bind with the cofactor. They are not yet activate. So therefore, they cannot bind with the substrate yet because this is a substrate, purple color. You can see that the active site is not fit with the substrate. So apoenzyme need activate so they can bind with the cofactor, okay, or the allosteric cofactor. Both when the cofactor bind with the apoenzyme, it form the holoenzyme. Okay, it form the holoenzyme. So holoenzyme is the enzyme which readily to carry out the catalytic process. You can see that there is two types of cofactor here. One of them is completing the active site. Another one of them is bind to the allosteric site, which lead to the activation of the enzyme to carry out the function. So this is the part where the active site is look like, the part that labeled in red color. So this is the active site. So normally during exam, if a question asks you to use an arrow to label the active site, so you can use the arrow directly pointed to any regions of the red color part to indicate that this is the active site, or you can also draw a column like this in order to label the active site. Okay, so once the active site is formed, substrate will possible to bind with the enzyme, forming the enzyme substrate complex. So in order to label the enzyme substrate complex, you have to bracket the whole thing. Okay, you have to bracket the whole thing. Bracket it, the whole thing, or circle the whole molecule to actually label it as the enzyme substrate complex. So after they enzyme bind with the substrate, okay, so the catalytic process is carried out, then the reducing of the activation energy is occurred, so it will lead to the formation of product. So the enzyme, the substrate will eventually catalyze to undergo the metabolic reactions, and the product is formed. So once the product is formed, it will move out from the enzyme, and the active site will readily to receive the new substrate to carry out the catalytic reaction. So at the end of the process, enzyme is not being consumed by the reaction. So that's it for the labeling of the enzyme.